Hello everyone. Today's topic is nutritional groups of microorganisms. And myself, Dr. A. Madhuri, Assistant Professor of Microbiology from Government Degree College for Women, Begumpet. Now, so the topic is nutritional classification of microorganisms. First of all, we have to understand why they are classified and how they are classified, on what basis. Basically, all the organisms require nutrients for their growth, and nutrient can be defined as any chemical required for growth of microbial populations. So the most important of these are carbon, oxygen, nitrogen, or hydrogen. So these are the four important elements which are required by the microorganisms for their growth. All microorganisms require source of energy and hydrogen atoms or electrons for their growth and development. In addition to these carbon, hydrogen, nitrogen, and oxygen, hydrogen, they also need energy sources and source of electrons also for their growth. Now, there are two sources of energy available to microorganisms, and based on this, they are of two types. Okay, so energy, how from where they are getting. If they are getting from light, they are called as phototrophs, and if the organisms are getting energy from chemicals by breakdown of chemicals, they are called as chemotrophs. Okay, based on the source of energy, microorganisms can be divided into two types. They are phototrophs and chemotrophs. Photo means light, trophy means food. In a food level, energy for growth is derived from sunlight. If the microbes are getting energy from sunlight, they can be called as phototrophs, and if they are getting energy by oxidation of either organic or inorganic chemical compounds, they can be called as chemotrophs. Microorganisms also have two sources for hydrogen atoms and electrons, and then based on that, they can be grouped as lithotrophs and organotrophs. So in the previous slide, we have seen based on the energy source, we have divided the organisms into phototrophs and chemotrophs. In this slide, we are classifying the organisms based on the source of electrons and the hydrogen atom. So here also microorganisms are classified into two groups. One is lithotrophs and the other one is organotrophs. So what are lithotrophs? Source of electrons is reduced inorganic compounds. If the organism is able to utilize reduced inorganic compounds as the source of electrons, then they are called as lithotrophs. Next category is organotrophs. Here, the source of electrons and hydrogen is organic compounds. Okay, so basically we can understand in simple terms that electrons and hydrogen atoms can be derived from two sources. One is organic compounds and the other one is inorganic compounds. If they are getting from organic compounds, they are called as organotrophs, and if they are getting electrons from inorganic compounds, they can be called as lithotrophs. Now, based on the energy, electron or hydrogen and carbon sources, microorganisms categorized into different uh, nutritional types. Okay, first one in this first one is photolithotrophic autotrophs. We can go with the names. Photo means light, litho means again inorganic compounds, autotrophs means they carry out photosynthesis. Okay, so these organisms are also called as photoautotrophs. Photoautotrophs. In short, photolithotrophic autotrophs can also be called as photoautotrophs. In short, as they use light, because in the word in the title, photo is there, so they are using light as their energy source, and carbon dioxide as their source of carbon. Example: eukaryotic algae and cyanobacteria, purple sulfur bacteria, and green sulfur bacteria. Okay, in all these organisms, photosynthesis will occur. That is why they are categorized into this category of photolithotrophic autotrophs. Here again I am revising. This is the first group. Its name is photolithotrophic autotrophs. In short, we have to call them as photoautotrophs. As the name photo is there, the energy is derived from light, and litho carbon, their carbon source is carbon dioxide, and as we can understand by the name, they carry out photosynthesis. So examples can be algae, cyanobacteria, and purple sulfur bacteria and green sulfur bacteria. In all these organisms photosynthesis will occur. That is why we are classifying these organisms in this category. Eukaryotic algae and cyanobacteria use water as electron donor and release oxygen. Purple and green sulfur bacteria cannot oxidize water and thus use the electrons from inorganic donors like hydrogen and hydrogen sulfide. In photosynthesis, there are two types: oxygenic photosynthesis and anoxygenic photosynthesis. What is oxygenic photosynthesis? Is when oxygen is released as a process of photosynthesis, as a byproduct of photosynthesis, then it can be called as oxygenic photosynthesis. In the same way, when oxygen is not released in the as by the end of the photosynthesis or as a byproduct of the photosynthesis, then it can be called as anoxygenic photosynthesis. Anoxygenic photosynthesis. So here eukaryotic algae and cyanobacteria, they are using water as the electron donor, and when they break down this water molecule, oxygen is released. Whereas in purple and green sulfur bacteria, they are not able to oxidize water, or they are not able to break down the water. Just they use electron donors as hydrogen and hydrogen sulfide. That is why oxygen is not released. Means this process is called as an oxygenic photosynthesis. Now, second category. First category, what we have seen is photolithotroph. Photolithotroph. Now, second category is chemo-organotrophic heterotrophs. Chemo-organotrophic heterotrophs. In short, they are also called as chemoheterotrophs. As the name shows, chemo means chemicals. They are utilizing chemicals. Heterotrophs means they cannot synthesize their own food, and they are depending on other organisms for their food. So with this term itself, we can understand this much information. So let us see what are these chemoheterotrophs. They use organic compounds like sugars and amino acids as source of energy, hydrogen and carbon. So as I said, these organisms, they cannot utilize sunlight, so they are using as energy source, uh, for the compounds. These amino acids or sugars are utilized as a source of energy and a source of hydrogen and carbon also. The vast majority of pathogenic microorganisms are chemoheterotrophs. So majority of the pathogens, whatever we are seeing in nature, they all come under, most of them, they are all come under chemoheterotrophs, because they are using chemicals for their survival, and they cannot synthesize their own food. That is why they are called as chemoheterotrophs, and most of the pathogens are included in this category. And the third category is photoorganotrophic heterotrophs. Now again, by the name we can understand that photo means they are able to utilize the sunlight, organotrophic means they are utilizing organic compounds, and heterotrophs, even though they are able to utilize absorb sunlight, they are not able to carry out any photosynthesis. They are heterotrophs. They are dependent on other organisms for their food. So some phototrophic bacteria like purple non-sulfur and green non-sulfur bacteria use organic compounds such as electron donors and carbon sources. For example, Rhodospirillum rubrum use succinate as electron donor. So here these organisms, They are phototrophic bacteria, um, like purple non-sulfur and green non-sulfur bacteria. They are using organic compounds such as electron donors and carbon sources. Okay, so organotroph term is there now, so they are using organic compounds, right, as source of electrons and carbon. Example is Rhodospirillum rubrum. It is using succinate as an electron donor. Next category, and the last category, is chemolithotrophic autotrophs. Chemo means chemicals are being used. Litho means inorganic chemicals are being used for their nutrition, and autotrophs means they are able to extract energy from chemicals. Okay, they are able to extract energy from chemicals. They are not dependent on any other organisms for food material. They oxidize inorganic compounds like iron, sulfur, or nitrogen to derive both energy and electrons for biosynthesis. As the name or the title is suggesting, lithotrophs means they are able to utilize inorganic compounds. Inorganic compounds means, example, iron, sulfur, or nitrogen. So these inorganic compounds are used as a source of energy as well as electrons. Okay, these inorganic compounds are giving energy as well as electrons for the microorganisms. Now carbon dioxide is a carbon source for them. Okay, so the carbon is taken from carbon source, and few chemolithotropes have been recognized to derive their carbon from organic sources, and they are called as chemolithotrophic heterotrophs. So in this category, we are seeing two types here. One is chemolithotrophic autotrophs, and the other one is chemolithotrophic heterotrophs. Okay, so as the name is suggesting, litho, they are able to utilize inorganic compounds as a source of energy as well as electrons, and carbon is derived from carbon dioxide. Uh, so these are chemolithotrophic autotrophs. But when carbon is derived from organic sources, then they are called as chemolithotrophic heterotrophs. Next is mixotrophs. Mixotrophs, the name itself showing mixed um, nutrition type is seen. Two types of nutrition is being seen in this organism. Bacteria that obtain energy by utilizing inorganic electron donors but obtain most of their carbon from organic compounds are called as mixotrophs. Here you can understand that these mixotrophs are involving both, or they are utilizing both organic compounds as well as inorganic compounds. Okay, here inorganic compounds are utilized as electron donors, and organic compounds are utilized for deriving their carbon requirement. So such organisms are called as mixotrophs. So in this chart, we can understand in detail whatever we have learned till now. We can sum up here. So energy source. If the organism is utilizing light, we have to write the term photo. Okay, if the energy source for any microorganism, if the energy source is light, then we have to write the word photo. If the electron source is organic, you have to write the term organo. If it is inorganic, you have to write the term litho. And coming to the carbon source, if the carbon is taken from organic sources, we have to call it as heterotroph, and if it is taken from carbon dioxide, we have to call it as autotroph. So these are the terminology which are being used to give the name or to classify any microorganism in the given classes or given categories which we have discussed till now. Okay, so let us categorize the microorganisms first. So first one is this entire thing is for phototrophs. Okay, so photo organo heterotroph means the organism is utilizing light as a source of energy, and organic compounds as a source of electrons, and same organic compounds as a source of carbon source also. So for this category, we have an example called as this nutritional type is called as photoorganoheterotroph. Okay, again one more revising again here. These organisms are utilizing light as a source of energy, organic compounds as a source of electrons as well as carbon source, right? So that is why we are calling it as photoorganoheterotrophs. Now next category is photoorganoautotroph. But in this category, there are no known organisms. So we are not giving any name for this type of microbes. And the third category is photolithorganotroph. Okay, here also organisms are not there in this category. And the fourth one is photolithorganotroph. Example is not example, they are called as photolithotropes. This is a category where photo means light is utilized as a source of energy. Litho means inorganic compounds are used as a source of electrons, and carbon dioxide is used as a source of um, carbon. So this we have seen in the previous slides. Example is rhodosmyrillum rubrum. And coming to the next category, that is chemical compounds, which we define by the term chemo. Okay, if the energy source is chemical compounds for any microorganism, we have to write or we have to mention it as a chemo. Okay, coming to the electron sources, again there are of two types: organic compounds and inorganic compounds. So here, Chemo-organotroph. Chemo-organoheterotroph. If the organic compounds are utilized as carbon source, we have to call it as heterotroph. If the carbon dioxide is used as a carbon source, you have to call it as a autotroph. So for the first category, we have to write it as chemo-organoheterotroph. Okay, which organisms are there in this category? And the second category is chemo-organoautotroph. But here in this category, there are no known organisms. And the third category is chemolithorganotroph. Chemolithoheterotroph. There are microorganisms in present in this category. And the last one is Chemolithotroph. Okay, so these are the categorization or classification of different microorganisms based on their energy source, electron source, and carbon source. We can write the what is the nutritional type of microorganisms based on these sources of microorganisms. Now let us study each category of microorganisms in detail. So for coming to the first category, first let us see about photoautotrophs. Here, these bacteria capture, as the name shows, photo and autotroph. Definitely photosynthesis is involved here in this category, right? So these bacteria capturing the energy of sunlight and transform it into chemical energy. In other words, what happens in photosynthesis? The microorganism or any organism, they will take up the sunlight and carbon dioxide from the air, and they will synthesize um, metabolic energy. Okay, so here also they are utilizing sunlight and they are transforming into chemical energy. In this process, carbon dioxide is reduced to carbohydrates. With the carbon dioxide which is absorbed from the atmosphere, it is reduced. What do you mean by the word reduction? Reduction means addition of hydrogen or removal of oxygen. So here to the carbon dioxide means here hydrogen is not involved. If you add hydrogen to this molecule, it will be called as reduced molecule, and then this will form a carbohydrates. This will form carbohydrates. The hydrogen donor is water and the process produce free oxygen. In photoautotrophs, definitely oxygen will be released as the electron donor or a hydrogen donor is a water. Now, chlorophyll is present in the cell and its main function is to capture sunlight. And one more important component required for photosynthesis is the pigment called as chlorophyll. Since childhood days, we are learned that chlorophyll is required for photosynthesis. Here also in bacteria also, bacteria chlorophyll will be there. So that is required for the photosynthesis. Example is cyanobacteria, and this is the overall equation for photosynthesis which is being carried out in bacteria. So here it is taking up carbon dioxide, water, and sunlight, light energy. It is converting this carbon dioxide to C6H12O6 carbohydrate or glucose, and then 6 water molecules and plus 6 oxygen. Oxygen is evolved in this process. That is why this process is also called as oxygenic photosynthesis, which can be seen in cyanobacteria or blue-green algae. The reaction produces free oxygen. We have seen in the equation free oxygen is released. However, in some bacterial photosynthesis, hydrogen donor is a substance other than water, hence oxygen is never produced. In the starting slides also we have seen when the organism utilizes hydrogen or hydrogen sulfide as an electron donor or hydrogen donor, they can't produce oxygen. So such a process is called as anoxygenic photosynthesis. This is called anoxygenic photosynthesis, and it is this anoxygenic photosynthesis is found in purple sulfur bacteria and green sulfur bacteria. Purple sulfur bacteria and green sulfur bacteria, they are showing anoxygenic photosynthesis, whereas cyanobacteria or blue-green algae is showing oxygenic photosynthesis. So here we have learned about photoautotrophs. These photoautotrophs are anaerobes and have bacteriochlorophyll and bacterioviridin pigments respectively. So these organisms, they are having pigments called as bacteriochlorophyll and bacterioviridin. In this process, these pigments absorb light and reduce carbon dioxide to form organic compounds. It is taken from atmosphere and hydrogen from sources except water. So in anoxygenic photosynthesis, they are having pigments called as bacteriochlorophyll and bacterioviridin, and they are taking carbon dioxide from the atmosphere and light from the sunlight, and the pigments will absorb the light, and they'll reduce this carbon dioxide to organic compounds, and carbon dioxide is taken from the atmosphere and hydrogen from sources except water. That is why oxygen is not released. If the water is involved in the process, then definitely oxygen will be released as an end product. But as water is not involved here, and it is taking hydrogen or electrons from other sources such as hydrogen sulfide or hydrogen, then oxygen is not involved and oxygen is not released at the end of the process. So in this purple sulfur bacteria, which is showing an oxygenic photosynthesis, they have the pigment called as bacteriochlorophyll, which is located on intracytoplasmic membrane, that is thylakoids. When we study the structure of chloroplast in eukaryotic cell or plant cell, we will come across this word called as a thylakoids. Okay. So in this bacteria also, that is a eukaryotic organism or higher organism, plant means. Here in bacteria also, these thylakoid or membrane, intracytoplasmic membrane system is present, where bacteriochlorophylls are located. These bacteria obtain energy from sulfur compounds, example chromachium, thiospirillum. So they are taking energy from sulfur compounds. For example, hydrogen sulfide. So carbon dioxide is taken from the atmosphere, and in place of water they are utilizing hydrogen sulfide, and light energy is as usual. So the reaction equation will be, in place of carbon dioxide they will be getting glucose, and in place of H2S they will be getting sulfur, and water is released, not oxygen. Now, coming to the green sulfur bacteria, these bacteria use hydrogen sulfide as hydrogen donor. Okay, in the last previous category also, we have seen H2S was being utilized. In green sulfur bacteria also, H2S will be utilized. That reaction takes place in the presence of light and pigment will be termed as a bacterioviridin or bacteriophyophytin or chlorobium chlorophyll. Example, chlorobium limicola, chlorobium, etc. So in the previous uh, category, we have seen bacteria chlorophyll is involved, but in green sulfur bacteria, the other pigments are also involved. They are called as bacterioviridin, bacteriophyophytin, or chlorobium chlorophyll. Now, these are the examples: chlorobium limicola, chlorobacterium, etc. So, due to presence of these pigments, these bacteria will be green in color as they are utilizing sulfur. Hydrogen sulfide, they are called as green sulfur bacteria, and this is the equation. As usual, they are taking carbon dioxide from the atmosphere and they are taking H2S plus light in the presence of bacterial chlorophyll. It will be converted into glucose plus 6 water molecules plus 12 sulfur. Here also, if you can see, water is released but not oxygen. That is why it is this process is called as anoxygenic photosynthesis. Oxygen is not released. These bacteria take hydrogen from inorganic sources like sulfides and thiosulfates. Therefore, these bacteria are also called as photolithographs. As there is the involvement of inorganic substances in this process, they are called as photolithographs. Next, coming to the second category, that is photoheterotrophs. Photo means light, hetero means they are dependent on other organisms for their food. These bacteria can trap light energy but cannot use carbon dioxide as their sole carbon source. So as the term photo is involved here, they are taking up the light energy. And as the term heterotrophs is involved, they are depending on other organisms. They cannot use carbon dioxide as their carbon source. They use organic compounds from the environment to satisfy their carbon and electron requirements. So in place of carbon dioxide, they are taking up organic compounds from the environment. So they use organic compounds such as carbohydrates, fatty acids, and alcohols. And the pigment is bacteria chlorophyll. Example is purple non-sulfur bacteria, which includes Rhodospirillum, Rhodomicrobium, Rhodopseudomonas, etc. And this is the equation. So coming to the green sulfur back, photoheterotrophs. In the previous slide, we have seen that these organisms, as the name heterotroph is involved, they are taking up organic compounds from the atmosphere. So, what kind of organic compounds are utilized by these organisms are carbohydrates, fatty acids, and alcohols. So, the pigment involved here is again bacteriochlorophyll, and the examples are purple non-sulfur bacteria, and this is the equation. So, this is the equation of this category, that is 6 carbon dioxide. They are utilizing organic compounds, and in presence of light they are making glucose and other compounds along with water molecule. Now, Chemoautotrophs, third category is Chemoautotrophs. These bacteria do not require light. As the term chemo is there, chemicals are being utilized and photo is not there. They are not able to absorb light. So lack the light phase but have the dark phase of photosynthesis. We know that photosynthesis is having two phases. One is light phase and the other one is dark phase. So in these organisms, we can see the dark phase but light phase is lacking. and pigment for their nutrition. So chlorophyll pigment is also lacking. These bacteria oxidize certain inorganic substances with the help of atmospheric oxygen. So they take up some inorganic compounds and they will oxidize these inorganic compounds by using atmospheric oxygen. This reaction releases energy which is used to drive the synthetic processes of the cell. Here, this reaction is releasing energy. Means they are these reactions are exothermic reactions. This energy which is released, it will be utilized for the processes of the cell. The source of carbon is carbon dioxide. In the absence of light, synthesis of organic food from inorganic substances by utilizing chemical energy is also known as chemosynthesis. Here, the source of carbon, the organisms are utilizing carbon dioxide. Okay, here the source of carbon is carbon dioxide, and in the absence of light, as the term chemo is there, so light they cannot absorb, and in the absence of light, synthesis of organic food from inorganic substances by utilizing chemical energy is also known as chemosynthesis. Means these organisms, they are utilizing chemical energy which is derived from inorganic substances. Okay, they are breaking these inorganic substances and they are getting some energy, so that energy is being utilized for the synthesis of organic compounds. So overall this process is called as chemosynthesis. The process is called as chemosynthesis. The chemoautotrophic bacteria play a very important role in recycling inorganic nutrients. As we have seen elemental cycles, that is carbon cycle, sulfur cycle, nitrogen cycle, all these cycles they are carried out by this group of bacteria, that is chemoautotrophic bacteria. These bacteria are commonly named after the structure of the compound which is utilized as the source of energy. So based on the compound which is being utilized by the organism, based on that compound, the organism name is given. So in this first one is nitrifying bacteria. As I said, based on the substrate which is being utilized, based on the compounds which are being utilized as an energy source, the bacteria are named. So here nitrifying bacteria. So what they are utilizing, these bacteria, nitrifying bacteria obtain energy by oxidizing ammonia into nitrate. That is why they are called as nitrifying bacteria. So nitrifying bacteria are converting ammonia into nitrate. The process occurs in two steps and each step is carried out by specialized group of bacteria. In the first step, ammonia is oxidized into nitrites by the bacteria Nitrosomonas and Nitrococcus. Okay, so the conversion of ammonia into nitrate is happening in how many steps? Two steps. In the first step, ammonia is converted into nitrite. First, it is converted into nitrite, and the organisms involved here are Nitrosomonas and Nitrococcus. And this is the equation: ammonia plus oxygen gives rise to nitrite plus some energy is released. In the second step, nitrites are converted into nitrates. Okay, example is nitrobacter and nitrocystis. So nitrite, what we have got from the previous equation, will react again with the oxygen and it will be converted into nitrate, releasing some more energy. These nitrifying bacteria are present in the soil and of considerable economic importance. These bacteria, which are converting ammonia into nitrates, they are usually called as nitrogen fixing bacteria, and they are of very much important um, to the nature. When these two groups of bacteria work together, ammonia in the soil is oxidized to nitrate is a process of nitrification. Energy released upon the oxidation of both ammonia and nitrite is used for chemosynthesis by these bacteria to make ATP by oxidative phosphorylation. So we have seen this, there are two groups of bacteria with two different equations. They are converting this ammonia to nitrate. Okay, so and we have also seen in both the equations some energy is released. So that energy is utilized for making of ATP by oxidative phosphorylation. So the conversion of ammonia to nitrate is called as nitrification. Next is sulfur bacteria. These bacteria obtain energy either by oxidation of elemental sulfur or H2S. Okay, here the term oxidation means addition of oxygen or removal of hydrogen. Okay, so these bacteria, what they are doing, oxidizing these elements. Which elements? Either sulfur or sul hydrogen sulfide. So elemental sulfur oxidizing bacteria uh, are denitrifying sulfur bacteria. Example, Thiobacillus denitrificans oxidize elemental sulfur to sulfuric acid. Thiobacillus denitrificans is oxidizing elemental sulfur means S, only S will be there to sulfuric acid. Sulfuric acid is H2SO4. And here also energy is released and this energy is utilized by the organisms for the chemosynthesis. So going with the equation, this is the sulfur in presence of water molecules. So it is reacting with oxygen and it is converted into sulfuric acid. Some amount of energy, that is 126 kilocalories, is being released. And the second step is sulfide oxidizing bacteria. These bacteria oxidizes sulfur dioxide H2S and release the sulfur. So in the previous equation, we have seen the organisms which are utilizing only elemental sulfur. In this reaction, we are seeing the organisms which are able to utilize hydrogen sulfide. Here, this example is Begiotova. When this hydrogen sulfide is oxidized in presence of oxygen, it is converted into water plus elemental sulfur. Here also some energy is released. Now, next category is iron bacteria. In this iron bacteria, they inhibit waters that contain inorganic iron compounds and oxidize ferrous compounds to ferric forms. Example, Thiobacillus ferroxidans, Ferrobacillus and Leptothrix. Okay, so iron bacteria, where they are present in the water sources or water bodies where iron is present. Okay, and there they are converting this ferrous forms to ferric forms. Ferrous compound to ferric forms. Ferrous means Fe2 plus, ferric means Fe3 plus. Okay, oxidation state of ferrous compound will be plus 2 and ferric compound will be plus 3. And the organisms which are responsible for this conversion is Thiobacillus ferroxidans, Ferrobacillus and Leptothrix. This is the equation. FeCO3, when it is oxidized, it will give FeOH3. Okay, and carbon dioxide. If you take the oxidation state, it is converted into ferric form. Next category is hydrogen bacteria. These bacteria oxidizes hydrogen into water. Okay, when hydrogen is oxidized means when hydrogen is reacted with oxygen, it will definitely give water molecule, and this reaction is catalyzed by hydrogenomonas, and this is the equation. Hydrogen, when it is reacting with oxygen, it is giving water plus 55 kilocalories of energy, and when four molecules of hydrogen are reacted with the carbon dioxide, it is giving carbon water plus methane. This is a methanogen. Okay. Next, carbon bacteria. These bacteria oxidize carbon monoxide into carbon dioxide. Example is Bacillus oligocarbophilus, Oligotropha carboxydovorans. These are the organisms which are converting this carbon monoxide into carbon dioxide by oxidation. Here, carbon monoxide is reacting with the oxygen molecule and it is giving carbon dioxide along with the energy. The last category is chemoheterotrophs. These chemoheterotrophs obtain both carbon and energy from organic compounds such as carbohydrates, lipids, and proteins. The carbon source as well as the source of energy are mostly the same for these bacteria. Okay, Chemoautotrophs, and the term itself is showing they are utilizing chemicals and they cannot synthesize their own food. Definitely, they are pathogens. Okay, they are their source of energy and source of carbon or the chain. Most of the bacteria are chemoheterotrophs. Here, glucose, they are utilizing carbohydrates, fatty acids, or other molecules. So, for example, if we take the glucose or monosaccharide, when it is reacting with oxygen, it will release carbon dioxide plus hydrogen, water plus energy. So, these chemoheterotrophs may belong to one of the three main categories that differ in how they obtain their organic nutrients. So, they will break down the molecules, biomolecules, and they will derive their energy. And these are, as I told, they are parasitic in nature. These bacteria obtain their food from living hosts on which they grow. They are pathogens or parasites. They will grow on another organism and they derive their food from those organisms. Parasites which cause disease are called as pathogens. Example, Clostridium, Mycobacterium, etc. Next category is saprophytes. The difference between pathogens and the saprophytes is pathogens are surviving on a living host, whereas the saprophytes they derive their energy from dead bodies. These bacteria obtain their food from dead or organic remains like fruits, vegetables, leaves, meat, fishes, corpses, and other non-living products. Whatever be the material, it should be in a non-living form. So that is then only we can call them as saprophytes. The anaerobic breakdown of carbohydrates is fermentation, while that of proteins is called as putrefaction. So whatever molecules when they are broken down in under anaerobic condition, when they are broken down, they will lead to the formation of the anaerobic breakdown of carbohydrates is called as fermentation, whereas anaerobic breakdown of proteins is called as putrefaction. So putrefying bacteria like Bacillus mycoides, Bacillus ramosus. Okay, so putrefying bacteria or protein degradation under anaerobic conditions is done by these organisms. Next is symbiotic. Symbiotic means living together. Sym means together. Bio means life. So symbiotic, these bacteria live in close association with organs of other organisms. Example, higher plants and animals in such a way that both the concerned organisms receive mutual benefit from their association. This is called symbiosis. Here, two organisms will be living together. They derive benefit from each other. That is why they can survive together. For example, here the microorganisms will get associated with animals or higher plants. Okay, so the best example for the symbiotic association is association of rhizobium and legume plants. Here, rhizobium fixes atmospheric nitrogen. It converts ammonia. It converts atmospheric nitrogen to ammonia, and this ammonia is given to the plants, uh, where it can form biomolecules from this ammonia, and whereas the rhizobium will get nutrients as well as shelter from the plant. So these bacteria fix free atmospheric nitrogen into nitrogenous compounds which are utilized by the plant. In return, plant provides nutrients and protection to the bacteria. In the stomach of cows and goats, bacteria digest cellulose, enabling these animals to feed on grass. Okay, we know that cows are herbivores, they feed on grass, but they do not have any mechanism to digest this grass. So in turn, some microorganisms or cellulolytic microorganisms will be present in their gut or stomach to digest the cellulose present in the grass so that they can feed on the grass. And the last one is mixotrophs. These they use different sources of energy and carbon. Possible combinations are photo and chemotrophy, litho and organotrophy, auto and heterotrophy, etc. And my organisms involved are eukaryotic and prokaryotic organisms, and it can be obligate or facultative. Obligate means compulsory nutrition, and facultative means it is not compulsory. So types of mixotrophy involves obligate mixotrophy. Obligate mixotrophy means obligate autotrophy with facultative heterotrophy. In this, autotrophy is compulsory, but when light is not uh, available, the organism will survive as a heterotroph. And the next category is facultative autotrophy with obligate heterotrophy. Here, the organism is actually a heterotroph, but when the source of nutrition is not available, then it can also survive as an autotroph for some time. And maintenance and growth may be obtained by heterotrophic and autotrophic means alone, and mixotrophy used when necessary. Now, uh, it is having only one type of nutrition, but when there is a requirement, it can change to another type of nutrition. So likewise, we are having methylotroph also. Here, single carbon compounds are utilized, and these are the examples. And they degrade methane, and the methane utilization. They are called as Methanotrophs. These enzymes are there, MMOs or PMMOs are present. They help these organisms, and these are the examples. So with this, can end this presentation. So we have learned about different types of nutrition present in the microorganisms. Thank you.